Hi, welcome to my lecture. I am Grissel Trujillo de Santiago. I am professor at the Tecnológico de Monterrey, Campus Monterrey at the north of Mexico. And well, please let me present you my group here in Mexico. We are the Álvarez Trujillo Lab in the School of Engineering and Sciences at the Tecnológico de Monterrey. This group of very talented students and graduate programs in the graduate programs of nanotechnology and biotechnology. And we have several postdoctoral researchers, undergrads doing volunteer stays with us. And well, Dr. Mario Moises Alvarez and myself are the PIs or the principal investigators of this team. And well, I'm sure that we share this big dream with many of you. We want to contribute with our little grain of salt in enabling this 3D bioprinting of full organs and tissues that can be used in clinical translation. And of course, we are not alone in this pursuit. Many important groups, many of you are working hard on making this dream come true. Okay, but first, this is a lecture on bioprinting, and we're going to talk about 3D chaotic bioprinting particularly. But let's define what is 3D bioprinting. And well, 3D bioprinting is an additive manufacturing technology similar to the regular 3D printing that extrudes or deposits materials layer by layer until a 3D object is obtained. There are printers of thermoplastics. Those are the most common ones, like this one in the picture. But also, there are 3D printers of metal, ceramics, cement, glass, and other things. And well, what makes a bioprinter is actually the material that we use. We need to use living materials, so we say that we are doing bioprinting. So there are different types of bioprinting, but the one that is used the most is bioprinting, extrusion bioprinting. And this one uses a biome that is commonly composed of a hydrogel and living cells. So this hydrogel is a polymer swollen in water, and this one is very similar to the materials in our tissues, so we can use them to culture cells inside these materials. So yeah, with 3D bioprinting, we can make a precursor of a tissue. So depositing layer by layer this bioink, this hydrogel containing living cells. And in that moment, we have a precursor of a tissue. It's still not a tissue, so we have to mature that construct so the cells can start to do their job, to replicate, to recognize the hydrogel surrounding them, to communicate with neighboring cells, and then eventually make this mature tissue. So I said that we use hydrogels. Using hydrogels makes a lot of sense because, as I said before, they are very similar to the materials in our tissues. Of course, cells are the building blocks of our tissues, but our tissues are not just made by cells, but also materials. And these materials are basically hydrogels. They are proteins, and they contain some very complex and beautiful carbohydrates like glycosaminoglycans, small molecules, and that very complex three-dimensional matrix contains a lot of water, so that could be named a hydrogel. So what we do with hydrogels when we prepare an ink is we try to emulate as much as possible this perfect extracellular matrix that our tissues have. There are many types of hydrogels. I'm just going to mention some of them because it's going to facilitate understanding the chaotic 3D bioprinting. One of them is GELMA. This is a photo cross-linkable hydrogel. Is a photosensitive hydrogel. So it's basically gelatin like the one that we eat at home, but it is modified chemically, so it can respond to light and make a liquid pre-gel, a precursor of a gel, a hydrogel, a solid hydrogel in a matter of seconds upon a stimulus of light. So when we prepare an ink, we have this gelma dissolved in PBS, in water-based buffer. And while it is still liquid, we incorporate the cells, we suspend the living cells, and then we can cross-link that hydrogel with this stimulus of light. It with alginate is very similar. This is another hydrogel that we use a lot, but instead of using light, we use divalent ions like calcium. So this alginate comes from algae, from brown algae, and they have this capability of making a pregel, a liquid pregel, into solid hydrogel upon the presence of ions. So we make ionic bonds in the three-dimensional matrix of hydrogel, and the hydrogel stays as a solid material. We can find many other hydrogels reported in literature that you can use depending on the applications that you are pursuing. But these two hydrogels are not only used by our group a lot, but by many other groups. In this review that we published recently, you will find some of the hydrogels that have been used most in the past 2 decades to make bioinks, and what are the cross-linking methods. And you can find the ionic cross-linking or photo cross-linking, temperature, enzymatic cross-linking, so on and so forth. So I recommend you to read this review paper, because it's like very easy to understand, and I think it's very helpful if you are starting in this area. So other things that gelma and alginate have, and I want to highlight, is that, well, gelma it comes from gelatin, right? And gelatin, it's a hydrolyzed version of collagen, one of the proteins that are most common in our tissues. So this gelma preserves some cell anchoring domains, and that is super important, because cells, the first thing that cells need to do to eventually make a tissue is to anchor to something. So if you are encapsulating these cells in this hydrogel, they have to recognize that cell, so that hydrogel, so they can attach to that, and then start to do more complex biological things that they know how to do. Alginate do not have that cell anchoring motifs, because it comes from brown algae, so it is not, they are not supposed to be there. But again, as I mentioned before, it's very easy to cross-link with divalent ions. So we take advantage of these properties, the ease cross-linking of alginate and the self-anchoring motifs of gelma, and we mix them together. We make interpenetrated networks, so we have both things, the easy cross-linking and the self-anchoring motifs. And we first cross-link with calcium, and then this helps or serves as a temporary template for gelma, just to be there when it is still not cross-linked. And then we apply the light stimulus, and then we cross-link gelma. So you have both polymers cross-linked in the very same construct. So this is just an introduction for us to understand the rest of the presentation, I hope. And well, again, talking about bioprinting, how far have we come? So excitingly, the first tissue, bioprinted tissue, implanted in a patient was already happened. This was an ear bioprinted with the cells of the patient, and this was implanted successfully to construct an ear. It was actually implanted in a Mexican woman, so this is very exciting, particularly for me, and I guess for Mexicans. And well, other great milestone that has, I think, motivated a lot of people that work in this area is what Professor Anthony Atala and his group in Wake Forest University already did several years ago. And it was actually not bioprinting, but they biofabricated an organ already in the lab. They were able making bladders in the lab. And they were successfully implanted in patients, and the bladders were functioning perfectly well. So this TED Talk is one of the greatest motivators in my career. So I encourage you to see it. And of course, I can't pass the opportunity to show you my selfie with Professor Atala. Okay, but in the own words of Professor Atala, of course, making a bladder in the lab is a great milestone, but there is still a lot of things to do when we think on more complex organs such as kidney, which is a solid organ, is not hollow like the bladder, and it has more complexity of in terms of architecture, the cells, the types of cells that it contains, the physiological things that kidney does. So just to have a comparison, no? I think it's very clear that if we just see these cartoons of both organs, we realize that it's much more complex to make in the lab a kidney than a bladder. So we need to make the field of bioprinting move forward if we want to achieve this, or a heart, right? So there are many challenges that we need to solve to really achieve this great dream. And the ones that we are focused on as Alvarez Trujillo Lab are these ones that you are looking at your screen. Our tissues are not homogeneous masses of only one type of cell or only one type of material. They are multi-material. They have this complexity of, are made of many materials and cells. Other things is that they are not just mixed or blended in an homogeneous way. They have this beautiful microarchitecture. Every one of our tissues and organs, they have a specific microarchitecture that is not only beautiful, but is functional. And that microarchitecture is very important for the functionality, the biological functionality of the tissue. And other thing is vasculature. That is one of the greatest challenges. There are many groups trying to overcome this challenge of making vasculature in a full organ or tissue in the lab in order to make bigger organs or tissues, because that's what we need to do if we want to print something big, because the cells that are inside the construct, they need access to oxygen and nutrients, and also a way to release other components that they secrete and can be harmful or toxic for them if they stay there. So we need to incorporate that vasculature if we want to print big and full clinically relevant size tissues and organs. And I don't know you, but I used to think about vasculature system like this, a classic book text picture, but it's much more complex than that, right? So our bodies are full of blood vessels. In this anatomical machines exhibition in the Chapel of San Severo in Naples, you can realize how complex is our vasculature. All that you see there as little brown cables are very well preserved real blood vessels in a human. So I think it's now clear that our body is replete of vessels. Every centimeter cubic in our body is full of blood vessels. So just to give you an idea of how complex this is, now how to make this in the lab. So today I'm going to tell you how we are trying to overcome some of these challenges, technological challenges, using chaotic 3D bioprinting. And well, to start the explanation of chaotic 3D bioprinting, I always use this example because I love coffee, and I think it's also helpful to understand how we make those structures in our constructs, because as it happens in coffee and cream, when you very smoothly agitate the cream in your coffee and you see those beautiful structures forming, those structures are being formed or be drawn, if you allow me, by flow, but by very, very slow flow. So we also use flows in chaotic bioprinting, similarly to this example that is drawing those structures. But instead of using cream and coffee, we use a suspension of cells and a hydrogel precursor like the one that I discussed before, a hydrogel precursor that is still liquid. So while it is still liquid, we make that structure with the cells, and then we freeze the structure, we stabilize the structure using, for example, UV light if we are using gel, or calcium chloride if we are using alginate, or both if we are using both, which is actually what we do. So that's the idea. We use flows to make a structure, and then we freeze that structure, cross-linking the hydrogel. And so with this example of the cream and coffee, probably you are thinking, well, yeah, but this is not reproducible. Every time that I pour cream in my coffee, I will see a different structure. And if we want to print organs and tissues in the lab, it has to be a reproducible process, and you are right. So for that, we are using, for making something that is robust and is reproducible, we are using chaos. And I know that using chaos for that is counterintuitive, is weird, because we normally use the term chaos to refer to disorder, but this has nothing to do with disorder or with turbulence. This is the mathematical chaos that I'm talking about. So this chaos has several characteristics. One of them is that it can create beautiful structures like these ones. And what I want you to see is that these structures are layered. It's made of many, many layers. Chaos is very efficient in making these layered structures. Unfortunately, these layered structures are very similar to the structures that we find in our tissues. Very often, nature uses layers for making functional structures, and our tissues are not an exception. Even at the nanometric subcellular level, we can find layers, right? Like in our reticulum, endoplasmic reticulum, or mitochondria, or other organelles. So yes, chaotic flows can create this complex structure. It is a deterministic process. Again, it has nothing to do with disorder. You can mathematically model chaotic flows and their outcomes. So actually what you are looking at your screen is the structure made with chaotic flows and the computational simulation, and you can see that they match, right? So this is reproducible, is predictable. And another thing that is very nice about chaotic flows is that they can form a structure in an exponential way. And this happens because with chaotic flows, the structure that we are making, remember, like we are drawing the structures like in coffee and cream, but those structures are being formed by flow, but by the formations of that flow by these stretching and folding actions, like bakery, right? When they do the 1,000 layers cake, they make these layers very efficiently by the formation of stretching and folding, stretching and folding. So in that way, you can make a lot of layers in an exponential way, because you are stretching and folding, you have 2. Stretching and folding, you have 4 layers. Stretching and folding, you have 8 layers, so on and so forth. With a regular recipe or regular flow, you will only have an extension of that structure in a linear way, not in an exponential way. So that is something very powerful of chaotic flows, that they can make a structure exponentially. So now explaining more precisely what we are doing now in the lab, we are using this chaotic printhead as a printhead. So you can think about this chaotic flow maker as a printhead that we use in a regular bioprinter. So here what you are looking at in your screen is the kinetics static mixer, and this mixer makes chaotic flows. So this doesn't rotate while we are using it. It is static. It stays static. I'm just rotating it for you to see how it's constructed inside. So this static mixer or this structure is housed in this pipe, and it is composed of several mixing elements. So here we have 4 mixing elements which are exactly the same, but they are located 90 degrees one with respect to the other. So we feed this extruder or this bioprinting head with 2 materials, and at the end we have a filament made of several layers of those materials that I infuse at the top. And those layers are intercalated, right? So for example, here, Carolina and Maria, they were using this syringe pump, these 2 hydrogels inks, just colored with fluorescent particles, just for the sake of demonstration. And then they used this kinetics static mixer. It had 3 mixing elements. So at the end, we could extrude meters and meters of hydrogel that contain these 1, 2, 3, 4, 5, 6, 7, 8 layers. 8 layers inside, intercalated layers, and the structure was exactly the same along the whole filament of hydrogel. And this is mathematically modelable, as I already said. This was a mathematical simulation made by Dr. Mehdi Samandari. So now I think you can imagine what's the concept here. So we use a kinetics static mixer as a bioprinting head that is making internal structure in the hydrogel filament that we are depositing for making a 3-dimensional construct with a Cartesian bioprinter, a regular Cartesian bioprinter. So we can make macrostructures because of the bioprinter, the regular bioprinter, but with control in the microstructure thanks to this bioprinting head that induces chaotic advection or chaotic flows. Just very briefly, I'm going to show you here one little video for you to see or to envision more clearly how is this process. This is actually wet spinning, just for the sake of showing the concept, but this is adaptable to a bioprinter. So here you see that we are pushing or extruding the 2 hydrogels of 2 colors, and the layers are being formed inside the kinetics static mixer, and we can print a lot of fiber, and you will see, well, you're just looking at the filament made of the hydrogel, and if we see the cross section at the end of the extruder, you will see these layers inside. And so you can also define how many layers you want inside your filament. If you can vary from having from 2 to 16, even more layers inside the hydrogel filament. And this can be regulated by the number of mixing elements that you have inside your extruder. This is, again, mathematically modelable. You can see here the simulation and the experimental results. Here, for example, we use 6 mixing elements that render 64 layers inside the filament of 1 millimeter. So you have a lot of layers inside a 1 millimeter filament that is going all the way through the filament. So here, just to test how far we could go in terms of resolution, we printed a filament 1 millimeter in diameter again, using 6 mixing elements, and with this we were able of accommodating 128 layers of roughly 8 micrometers width. So this resolution, 8 micrometers, is not doable with other extrusion bioprinting techniques. Actually, here we had to use very small cells, bacterial cells, because they are smaller than mammalian cells, just to show that we could reach that resolution. Here is already with mammalian cells with myoblasts, which are precursors of skeletal muscle tissue. And we wanted to see if the cells were alive after the extrusion and during a long period of culture. So we cultured these constructs, these filaments with layers inside, and we were able of seeing high viability of our cells after 28 days. So this means cells are alive and kicking. The process is cyto-friendly. And this makes sense because normally when you want to increase the resolution of your bioprinted constructs, the first idea is to reduce the nozzle so you can have finer features in your printed construct. So, but this is not very useful for cells or just useful for bioprinting because cells are very sensitive to high shear stress. So they are going to die because if you decrease the nozzle size, you increase the shear stress substantially, and the cells are going to die. So cells prefer a wider nozzle to be extruded. So here in 3D chaotic bioprinting, the resolution, meaning the size of the features that we get, is not a function of the diameter of the nozzle, but of the number of mixing elements that we use. So you can accommodate a lot of very fine features inside a filament of 1 millimeter that is huge for the cells. So the cells do not even notice that they are being extruded. They are just being like accommodated by the flows through the bioprinting head. So with this, we show that cells are alive after our bioprinting, but also after several days of culture, we saw the cells exhibiting signs of maturation. So what you are looking at here is a close-up of cells that were printed with our chaotic 3D printing, and these were myoblasts. And when myoblasts start to mature by themselves, they make this actin or cytoskeleton made of sarcomeric alpha actin. So at the beginning, they didn't have this sarcomeric alpha actin, but after several days of culture, if they are not only alive but viable, if they are healthy, they start to make these structures inside themselves. So this is the sarcomeric alpha actin revealed by immunostaining, and the nuclei of the cells stained with API in blue. So these little filaments that you see are myofibrils that are present in skeletal muscle tissue. It's not a perfect mature skeletal muscle tissue, but cells are exhibiting signs of maturation. Also, with the internal structure that we can make in the cells, we can tell them what or guide them. For example, in skeletal muscle tissue, it is very important for cells to align. So the alignment is very it has a close relation with the contraction of the skeletal muscle tissue. So here what we did is to place layers of alginate that is not responsive. Cells do not respond to alginate. They cannot attach to alginate. And the orange layers here are representing a bioink that the cells can attach to, like GELMA, and they were in those orange cells. So or those orange layers. So the cells, what we are telling them to the cells is you have to follow that path that I'm telling you. So you cannot cross the alginate. So you have to align towards the main direction of the filament. And that is exactly what we observed when we make this design inside the structure of the filament. So this is exactly the same materials, but we didn't have that structure. We didn't have that layer structure. So the cells remain rounded, spheric, and they do not align. So even the cells inside, the alpha sarcomeric alpha actin is also aligned towards the main direction of the filament, which is very exciting. So I am running out of time, but I want to show you what we are doing regarding vascularization. So we are using also a scanning static mixer. But we are playing with the materials or the inks that we are using. There are some materials that can be cross-linked, like alginate, as I already explained. But other material is not going to cross-link, is not going to make a solid hydrogel. So it's going to remain liquid, and it's going to leave the hydrogel structure, the filament made of alginate and gelma, and it's going to form channels inside, hollow channels inside, because it remains liquid, right? So in this case, we use a combination of alginate and gelma, and as a sacrificial material or fugitive material, pluronic or HEC, the hydroxyethyl cellulose, the one that is not going to cross-link and is going to form these channels inside. So here I'm showing you how we were able to make these hollow channels inside, and we show these that they were actually hollow when we perfuse green particles in our filament, just for the sake of demonstration that they are actually hollow. Here in this scanning electron microscopy, Carlos and Joanna were able of making these hydrogel structures with a lot of empty channels inside. They are very ambitious, so they wanted to push technology the most they could, and they were able of accommodating in a filament of 1 millimeter in diameter, 32 empty channels inside that hydrogel. So this is already very close to that body that I showed you before, right? That a body that is really replete of blood vessels. This is still not not yet blood vessels, but these are already empty channels that are running through a very soft filament made of hydrogel. So very briefly, yeah, you can read this in a paper that is already published, but we were able of putting in our permanent hydrogel, the hydrogel that is making actually the filament myoblasts. Again, we are very interested in skeletal muscle tissue because of several reasons. One of them is that we are making meat for eating. We have launched this startup called Forma Foods, so you can look for Forma Foods, and we are also interested in skeletal muscle, not just for biomedical applications, but also for cellular agriculture. So here, coming back, we made a filament made of cells and these empty channels, and the cells that were printed with this architecture that contains the channels, they were more happy. The viability was higher, especially at day 7 of the culture that we follow up, and the metabolic activity was also significantly higher in the cells that were printed with the filaments with channels. Also, we assessed hypoxia markers and proliferation markers, and the hypoxia markers were underexpressed in the filaments with channels, as expected, of course, because they have more access to nutrients and oxygen than those that didn't contain the channels but were solid. And also the proliferation markers went up when we have these empty channels. So here you can see also the cells are aligning towards the direction of the channels, and they are growing in the whole filament because they have this access to nutrients and oxygen because of the channels that we were able to accommodate inside. Also, the markers of that we expected to see as signs of maturation of skeletal muscle formation, they were present. The alpha sarcomeric actin was there, and we observed this by immunostaining, but also by PCR. So I'm just going to close this presentation with a sneak peek of a paper that is coming soon published. It was recently accepted, but we were able of showing that we can print multi-material, not only 2 materials as I have shown in this presentation, but 8 different materials, right? And this opens more possibilities. We can make more complex architectures and more relevant. For example, talking about skeletal muscle tissues, something that we are doing now is some of these inks are going to be the skeletal muscle, some others are going to form channels, and some others are going to make an endothelium surrounding those channels to make a vascularized tissue closer to the tissue or muscle that we have in natural systems. So this is a versatile tool. We have shown its application also for culturing cells and looking at how these different bacteria compete or help each other depending on the architecture in which they were printed. We can also adapt this to electrospinning setup to make very fine nanoscale fibers with internal nano architecture that can be used for sensors, for example. And this is a very cost-effective tool. You don't have to invest $40,000 for buying a bioprinter, but you can adapt this to a regular setup of a bio or a 3D printing that you can adapt for making to print hydrogels and make a complexity with simplicity, complex structure with a very simple process, as I showed already here. So with that, I want to acknowledge all the students that were participating in making these results that I showed you today, and to my collaborators and the agencies that funded these projects. And thank you. So I hope that you enjoyed this lecture. I hope that it has been clear enough, and I'm looking forward for your questions. Thanks a lot.